Alright boys, Assalamualaikum warahmatullahi wabarakatuh. Alright, please sit down, lower down all the chairs. Now, right, for today's class, okay, please don't touch the devices just yet. Okay, for today's class, you're gonna have to work with a partner. Who's absent today? In total, how many students are there? 19? Right, 19, who's absent? 19, 20, 21. Ashraf, Daniel, and? Okay. Right, so meaning 19 of you, there should be 9 doing it with a partner, 1 student doing it alone. Right? So move on, listen up for my instructions, hold on. Move on to where the microscopes are. There's only 2 here, so there's going to be 2 groups sahaja di sini. Okay, there's 3, 2 there, and another 3. So move on, move along. I need 1 group, gotta move forward. 1 group, siapa? Okay, bah. Move, shift to the side. Bah, mana? Okay, ready? Tegar seorang kah? Tegar sama Lut? Nasharang. Ani, where you going? But you gotta move, Ishraf. Ishraf, you gotta move there. Yeah. Ani, complete. You're working with who? No, Ishraf. Gotta do it there. It's a Nash seorang. Right, now that's out of the way. Can we get started? Can we get started? All right, please make sure that there are devices in each bench, right? I think there's a laptop here, tablet, phones at the back, right? Para tegar. Right? Okay, so those devices, you're going to use it within your bench. But most of the time, you're going to work with your partners, right? So now, let's start. Everybody ready? Okay. Hold on. Okay, so that's slanted. Okay, so now, today's class, we are going to talk about the movement in and out of the cells. There's going to be 2 major lesson objectives, 2 goals of today. Here, sit down, right? So now, 2 goals. Number 1 is to identify and label key structures of plant cells. Okay, these are the ones that you have been identified with since last year, early of the year. Okay, and you're going to observe under the microscope, especially goal number 2. Goal number 2 is for you to be able to predict. Predict means guess. Okay, and observe changes. Changes meaning perubahan, right? In onion cells during experimental procedures. Okay, so these are the 2 goals that I want you to achieve today. What's the moral integration? I know you'll be working with a partner within your bench, but I want everybody to collaborate. Collaborate meaning that you're going to have to work together. Okay, each one of you is going to say different things than your friends, but please be respectful of the differences that you have. Okay, I know you're going to say one thing and you think it might be wrong what your friend is saying. Be respectful, value the different perspectives, and let us all, including myself, have open-minded discussion of what you're going to observe in today's lesson, right? Now, as a lesson starter, what are you observing here? My request is just one. Look at the middle of the bench. There's a small whiteboard, mini whiteboard, with a marker. Can you please tell me which bench doesn't have a marker? All right. I am showing you, what am I showing you here? Your first lesson starter. Plant cell, very good. Okay, now since you answered plant cell, can you tell me why do you know that it's a plant cell? You're 100% correct, but can you tell me why is it a plant cell? Why didn't you say it's an animal cell? Regularly shaped, very good. Other reasons? Huh? Cell wall, very good. Okay, now my request is in each bench, now as a big group first, right? Please draw just one cell. Goal number 1, identify and label key structure. Okay, draw just one cell from this picture and label the cell organelles that you see. Less than 3 minutes starts now. Draw on the mini whiteboard. Work within your bench first. The question is, okay, everybody listen up. For those who didn't get the question, draw just one cell from the diagram and the cell organelles. Label the cell organelles that you see. Very good. Mashallah, songkok. Songkok pakai. Very good. Right, now, I think everybody's almost complete. I want one representative to just raise the whiteboard. Okay, just raise it so that everybody can see what you have written down and what you have drawn. Lut, angkat saja. Hah? Pandang. I say label kan? Nash, angkat. From that bench, can you raise the whiteboard? Right. No changes after this. We'll wait for Mukri punya group. Okay, Mukri. Okay, one more label and then raise it up. Okay, now I want each bench starts to observe what you have written down. Which bench is different than other benches? Very different. Can you raise yours? Mukri tinggi-tinggi. Which one is very different compared to the other benches? This one, why you guys saying this one is different? What's different than the rest? Yang ni? They noted down chloroplast. This one? Luth punya group? Cytoplasm, cell wall, nucleus, cell membrane. Nash punya group? They label nucleus, ribosome, sap vacuole, cell membrane, cell wall. Whereas Mukri's group, cell wall, nucleus, cell membrane, and cytoplasm. So which one is actually correct? All is correct, but back to my question. I ask you to draw a cell, one cell from what you observe here. Can you see the sap vacuole? Can you see the ribosome? Can you see mitochondria? So Mukri's group, why are you drawing mitochondria? Why are you drawing sap vacuole? Okay? There's a lot of things that are differing. This will be very crucial when you want to draw to the activity after this. Okay? So which one is the closest that I want to see? It's actually from Lutz's group. Right? Okay, clear on the lesson starter? So now, here, before we move on, now you're going to work with partner. The first thing that you're going to do, you're going to prepare your microscope ready. Not now. Listen to my instruction first. You're going to prepare your microscope ready. You're going to predict the results. Now, what happens during prediction, boys? What happens during prediction? Prediction is when you guess what you are going to observe. Okay, I will tell you first what you're going to observe. Now, how do you make the predictions? It's going to be an individual thing. Each bench will have the devices, laptop, tablets, and phones. Phones is here. Later, I'll give it back to you. Okay, and then you will be able to access Padlet. Each person will submit their own prediction. Write down your name, write down your prediction. Doesn't have to be long. 3 words is enough, not sentences. 3 words is also enough. We don't have enough time, right? And then you're going to submit. And then you're going to see the specimen using the microscope after the prediction. Then you record using the worksheets I'm going to give you. And number 5, you're going to explain. Okay? Ready? Now, this is the prediction. Use the devices on your bench to predict what will happen when, this is the question, when an onion epidermis is immersed in concentrated salt solution. Right? Use the devices that is being prepared in each bench. Okay? You may start now. Okay. Write down your name, write down the answer. There's only less than 10 minutes. Type. Everybody has to answer. Individual thing kan? Okay, press. Yes, individual. So you're going to submit from that bench, you're going to submit 4 times lah, huh? Right? Yes. Yeah, the cell itself. Like what happens if I have a container, I have a beaker with salt solution, let's say, and I put the whole onion in it, and I take one cell, one skin of the onion. Yeah. Everybody should be able to start answering out from other benches while I get this one to work. All right. Now somebody's answering. Right. Yeah. It should only be very simple. Okay. Siapa belum? So mesti you have to submit 3 kali lah ha. After Nash and then tekan publish.
Jadi sharing kan?
Okay. Tekan publish.
Shuling ka verse lang.
It should be an individual thing. Mukris and Adam, different.
Shuling.
Should be an individual thing. After Mukris answer, and then publish. Tekan publish.
Zed.
All right. And then another one.
Shuling thing mo. Shuling nga. So pinalak. Sa ako, sa... Hindi yung nag-alba eh. Ano tayo ah?
Finish all three? Hello? Yeah? Regardless dah. Yeah. One person submit. And not satu answer. Like let's say Tegar write down his answer and then tekan submit. Left answer, tekan submit, and so on. Have you done all four of you? Belum. Less than 5 minutes. Sudah all three? Okay, give me. I have an answer. Okay. Nah. This one complete? Complete? All four of you? Seorang lagi. Ini tinggal seorang last. Okay. Nah. Boys, I don't want you to erase from the mini whiteboard. From the mini lesson started just now, do not erase what you have drawn. Okay? We're going to get back to that lesson starter again. Done? All 6 of you done? Okay, thank you. Clear? The last bench? Done? Okay, you may switch off the tablets, right? Switch off the devices. Okay, switch off the tablets, boys. Okay, may I know which bench are not done? Done with this part? All right. So now, let's look what we've seen here. Now, predict what will you observe when onion is immersed in concentrated salt solution. We know for sure that something's gonna happen, right? Oops, sorry. So now, let's say, take Mirul's answer. Right, onion will shrink, Nash, onion cell will shrink, Zaid, lose water due to diffusion, onion cell shrink, lose water, shrink, lose water, shrink, cell diffuse, shrink its size. Shrink, lose water. Irfan, shrink. Okay, a lot of you are giving shrinking, right? Now, good news. Shrinking is the correct term that you're supposed to use. But before shrinking is going to happen, there's a stage that nobody has given the answers here. There's a stage that's happening before shrinking. Very good, water move out of the cell, yes, but what do you call that cell? After water move out from the cell, what do you call? Not flaccid. I hear the word plasmolyzed. Right? Nobody is talking about plasmolyzed here. That's supposed to be included in your answer. Right? Now, if I reverse the question, what will you observe under the microscope if the onion is immersed in distilled water? Are you going to observe plasmolyzed cell? What's going to happen? Bukan placid. Osmosis is going to happen, yes, but turgid, very good, excellent, right? The answer is turgid. But the specimens that are prepared for today, it's at the middle of your bench, at the slide, right? Okay, you just take one for your own, together with your partner, one, and then put it under the microscope, and I want you to start observing. Now, wait for my instruction, boys. Here, okay. Your onion epidermis, your specimen is being immersed already in concentrated salt solution. I only want you to use the microscope and observe what you saw. This is in your worksheet question number 1, which I will distribute. 10 minutes, it's running now. While observing, I want you to draw immediately and also goal number 1, label. Of course, it's already being immersed in salt solution. There's going to be changes from the lesson starter, correct? Okay? So, what I want you to do right now is start using the microscope and draw what you observe. Turn on the microscope and start answering from the worksheets. Okay? Anybody who needs help, please raise your hand. You can take from the box. Yang ada wire, you can take from the box di belakang. Always start from which lens? Smallest lens, very good. You don't start immediately from the biggest lens. Okay, thank you. All right. If you cannot see anything, which knob do you adjust? Very good. Please write down your name, class, day, and date on the worksheet. I will have to get it back from you by the end of the lesson today. Yes? Very good. That's the basic practical. Draw using pencil. Label using garisan. Write down your name, class, day, and date on the worksheet.
Sebenarnya itu apa? Yang pasar, wah. Untuk taruh punya image. Nampak?
Please raise your hand if you need any help. Dapat? Nampak? Let me see. Ni, all the things that you see here, semua ni cells ni. Can you see? So now, all those things that you see, the cells start moving here. Okay? Now, it's very clear. You have a very good specimen there. Yes, boys, read the question carefully. You are only required to draw how many cells? One. You give me millions of cells, I'm not gonna mark it correct, huh? Even all labels are correct. Draw and label. Can you see something? Nash, you're doing it alone. Can you see? Coba, let me see. What are you looking at? Nampak kan tu? You're towards the end. No, it's supposed to be in the middle lah. It should fall on the specimen. Yeah, look at here. This one ada bubbles, but you can see cells here. Look at all of this are cells. Now, Chuba try moving it to the bigger magnification. I want you to find a cell that actually shows a nucleus first, so that it's easy to differentiate. Now, Nash, tell me, from what you draw here, lesson started, and from what you observe, do they look the same? No. What's the biggest difference that you can observe?
We can't see the small such as ribosome.
Tapi tadi pun ada kan, pada lesson started ada kan ribosome. Try to find out what is the biggest difference that you see. What's the rule? What's the rule of drawing? You draw it big using the space provided. No feathering, no coloring, no shading, solid continuous line. If I see correct labels, but a lot of shading, a lot of feathering, I'm gonna mark you wrong. Hey boys, we're gonna start drawing. What's your paper? Everybody has to have their own paper. Yes? That's right. Look. Name, class, day, and date. Less than 5 minutes. Ishra. Name, class, day, and date. Let me see what you're looking at. Let me see. Have you gotten the cells?
Yang online, si Macron.
Ni? Ini banyak kan?
Sila
Okay, let me see. Nampak?
Kami je. Ini yang spesifik. Sandu lah.
Ni nampak je ni.
Aku nampak nak kuning je yo. Gara.
Oh.
Apa ni?
Cuba kau main numpang ni.
Not at all. Ini apa? What is this? Cari satu-satu lah, benar kan tuh? Okay, now. Ini tiba-tiba ada benar tinggal. Nah, this is a very good example. This is what I want you to observe. Very good specimen. Look.
Mau cari lagi.
Nih.
Cari lagi.
Apa? You know, each person has to draw on their own.
Ngomong gitu. Ada ucapan. Baik, tapi lihat. Tadi je kan. Aku nak ikut berapa? Ngali pai. Ada pun lain. Kawan ni dah.
Very good. It's much bigger now.
Oh, apa? Tapi saya.
Very good.
Ibu nampak dua ni. Aku nampak sepuluh lagi. Aku nampak 5 nukleus.
Name, class, day, date. Write it down.
Oh.
Side. Do you need more time?
Yes.
Okay, I'll give you. Okay, the timer's got stopped, but I'll time you again here. Okay, 3 minutes.
Available. Saya penuh kesulitan. Kan bikin-bikin. Lukis itu. Ada semua. Tapi lukis kan ada. Kamera apa? Revolution. Yoyan. Oh, ada lukis. Revision mo kasi. Yes, ni bitin-bitin aja. Yang siya.
Eh, stop.
Bukan masalah video. Kami video.
Label.
Wala mo bawahan.
Label.
Revision mo.
Apa? Ilang mo kita. Remember, when you cannot see clearly, you gotta play around with this knob. Okay? Nah, adalah. Ada.
Langsung yang isinya. Oh.
Okay, while waiting for the others to finish drawing and labeling, you may proceed to question number 2. You should be able to answer. This is the explanation part.
SMS ka.
Very good, Lud. Good. Start answering question number 2. Ryan? Number 2.
So ano na? SONES. SO?
Yes, answering question number 2.
Tapin, bulag mo siya. Tapin, very good. Tapin, bulag mo siya. Ano yun? Mo.
Yes, Mukres. Yes.
Becomes ring.
Now you gotta figure it out on your own because we have covered this during the lesson. You have to label, Mukres. Yes, you gotta label. Goal number 1: identify and label. Okay, this is the explanation bit. What do you think happens when onion epidermis is immersed in concentrated salt solution? This is question number 2. Everybody should be able to answer this because you have covered this early part of the lesson. Okay, let me see your slides. Let me see your slide, what you have been observing. Because you're using the smallest lens, kan? It would be nice. It's okay, you don't have to redraw. It would be nice if you get a closer look, a bigger magnification. Look at this particular side. Okay, Ryan, can you look?
Small. Become shrink. Rasa di ba ni shrink? Rasa shrink ni sini.
Okay. Yes? Ini lupa lukis lagi. Hold on, let me see. I want you to be able to. Okay, where is it?
Ay itu do.
Now, look. Okay. Hello. What is this? Very good. Sawo. What is this? Very good. Yes. What is this? No? Cytoplasm part. Excellent. So, if you, because your label here is correct, cytoplasm. So, looking at it here, because I told you, you guys punya bench tadi close enough what I wanted to see, kan? You have nucleus, you have cytoplasm, you have cell wall, you have identified here. But this is your lesson starter. If I ask you, is your lesson starter, your observation similar to what you observe now? No, not really, right? So now, you got all those, but now the question is, where is the cell membrane right here? Can you point out where is the cell membrane? Point it out with the pencil. Where's the cell membrane? In this one cell. Yeah.
It's like very close to the cell wall.
No. Where is the cell membrane? Which one? Anis, this is cytoplasm. Right, very good. But now my question is, where is the cell membrane? Hindi kan cell membrane niya hilang. Right.
You know, like
Which one, right?
this one.
Excellent. So now, during your lesson starter, your cell membrane is so close to your cell wall. But now it is as if what is happening to the cell membrane? It got pulled away, right? So this is without salt solution. But now we have immersed it in salt solution. So now the question is there a change? Yes. So the change is in your question number 2. Should be able to explain that in terms of question number 2. Okay, let me see. What you have observed? Which one has the best specimen view? Let me see. This is very good. This is very excellent. I want. Very good. Good job. Now, I'll send the picture later. Where is it? Very good. Okay. Is this the same image as you see during the lesson starter? Lesson starter. Ada yang sama. Okay. So what are you looking at now? So if I were to label, let's say this one particular. Tadi kamu lukis cuma ni. Cell membrane, cell wall, nucleus. Let's take this one cell. Mana nukleusnya? Point it out using the pencil. Very good. Where's the cell wall? Very good. Where's the cell membrane now? No? Where could the cell membrane? Did it disappear when it is being put in salt solution? No. It shouldn't disappear. It shouldn't. Mana? No. One more try. It's okay. We're learning here. Okay? The more mistakes you make, the more you learn. Okay? Which part? Yang mana? Okay. That's the cell wall. Which one? Very good. Mana? Very good. Mana? This is your cell membrane. Pasal? Very good. So being put in a concentrated salt solution doesn't make the organelles disappear. Okay? It shouldn't do that. Okay? But it causes changes such as shrinking, which you have already covered in lesson starter tadi kan? You predicted tujuh tadi, shrinking. Okay? So here, where's the shrinking part? There. So now question is there any change? Okay? Okay. Now, let me see. Mana? This particular slide? Sini. Let me see. No, yang ni is very good. Let me see. Zoom in. Yeah. Let's magnify it a bit. This one is very good one. Anyang, what I actually want you to observe. Eh, this one. Yang ni kamu draw ada cell wall, cell membrane, cytoplasm, nucleus. Now, Ishraq and Akil, now tell me, mana cell wall here? Point using the pencil. Mana cell wall? Very good. Very good. Mana nucleus? Very good. Where is the cytoplasm? Very good. Now question is, where's the cell membrane?
Panglukis kan red blood cell kan?
Very good. Bukannya di sini lagi kan, bukannya ambil cell walls. Yes. What is happening to it?
This is goes away from cell wall.
Very good. It goes away. It got pulled away from the cell wall. That's the shrinking process. That's the plasmolysis stage. Now question is there really a change now compared to kalau ia ingat ditaruh hara salt solution? Yes. Okay. Question number 2. All right, boys, we're nearing towards the end. I think everybody has obtained a very good specimen and a very good drawing. I will need this paper back up. Now, let's move on to the lesson plenary, last one. What do you think will happen when an animal cell, such as red blood cell, is immersed in the same solution? Anybody has a good guess? Raise your hand. The specimen is being put in concentrated salt solution, but this is plant cell. What if I put an animal cell, such as a red blood cell, in the same concentrated salt solution? Give a guess. What's going to happen? Yes? It will?
It will shrink and be plasmolized by
By
the process of diffusion.
the process of?
Diffusion.
Not diffusion. That's when you are wrong.
Osmosis.
Osmosis. Because we're referring to the movement of water. Good try. But also, I want to correct on that part. Okay? Because you're putting it in concentrated salt solution. Meaning, water potential will be higher inside or outside the cell. Again, I repeat. If I put red blood cell in a concentrated water solution, which one has a higher water potential? Inside the cell or outside the cell? I repeat, red blood cell put in a container of concentrated salt solution. Which one has a higher water potential? So movement of water should be inside to outside or outside to inside? So what he said just now, it will shrink. It does not shrink. Instead, it will lose water. Water will move from outside to inside. It will shrink, but other word than shrink. You gave this word just now. Not turgid, not plasmolized. Remember, the word plasmolized only applies to plants. It is placid. Okay? Now, one volunteer from this bench, Mukres punya bench, can you read back your question number 2? Complete sentence. Yes. In pure concentrated salt solution, what happens? Mukris. Okay, everybody listen up. When one person speaks, everybody listen.
In the concentrated salt solution, the onion cell becomes plasmolized because water moves out of the cell by the process of osmosis. The cell vacuole becomes small in size. Water molecules move from high concentration to low concentration through partially permeable membrane. Concentration gradient.
Everything is correct except the last one. Good, very good job. But the last one is wrong.
Concentration gradient.
Blank concentration gradient. What's that supposed to be? Not high concentration gradient. Not low concentration gradient. Movement from high to low. There's a term for that. Starts with A. Ends with G. Starts with A, ends with G. A long concentration gradient or down concentration gradient. Okay. So question number 3. What happens when an animal cell is immersed in the salt solution? Water will move out of the cell, yes. What do you think will happen to the structure? You may draw in question number 3. What will happen to the red blood cell? Right, I'll give you 2 minutes and then you may submit the paper to the front. That's the end of today's lesson, right? Please submit back to the paper once you are done to the front. Okay, I need your help. Once you are done submitting the paper, all the microscope slides, put it in the middle of the bench. Microscope, put it back where it belongs. The plugs, put it back in the box. Once you are done, before you leave, please put back the chairs on the bench. The plugs in the box. Please carry the microscope carefully. Slides. Slides put here. Microscopes are not. Mini whiteboards, put it at the front. Yeah, it's okay. You don't have to erase. Right. Thank you. This all the microscope up. Okay, microscope. Apa? Yes. Alright, thank you boys. Assalamualaikum warahmatullahi wabarakatuh. Alright, thank you boys. Please put back the chairs on the bench. Alright, you may leave. Please don't leave anything out. I will lock the lab. Okay, thank you, Shah.